Hi class, I'm Professor Popowich. I hope that you had a good first half of forensic anthropology and bioarchaeology. I'm picking up the second half of this course, and today we're going to start to talk about estimating sex. So we're going to begin with an introduction, inquiring into what sex is from an anthropological perspective and what we can possibly know and some of the problems that we face. And here I'm using a word very carefully. I'm not saying we're determining sex. In various number of situations, we say that it's indeterminate. So we prefer to use the word estimating. That's what we're doing here. And we're going to be looking at a few individuals here. If you can see this individual here, we're going to inquire into whether or not this individual is male or female. Like I've said, we're going to start out with an introduction, and this will be a three-part lecture. So an introduction, then we're going to look at the skull and how we estimate those features. We'll have an example. And then we'll move on to perhaps the holy grail of estimating sex, which is our pelvic girdle or the two innominate bones. They do provide us with the greatest amount of information when we're trying to estimate sex. The other thing to keep in mind is when we're estimating sex, we're doing this on post-pubescent individuals. Very difficult to determine sex on individuals who are not or who haven't gone through puberty, because we're not seeing those changes in their bone as of yet. That eventually takes place. So here we're talking about post-pubescent individuals, adults. Okay, let's move on to the next slide. Okay, I've disappeared if you're wondering where I've gone. I just take up too much memory, so if you're looking for me, I'll put myself over here in the corner, right? There I am, Prof. Pop. I'm still with you. Here we're looking at a case called the Harper Road Woman, and this particular set of remains, they were discovered in London in 1979, and they haven't really undergone a whole lot of analysis up until recent times. So as pointed out, they're excavated in Southwark, London, in England, and these represent ancient Roman remains. So this is one of the first occupation of Roman individuals in England. And as for a calibrated date, so I know that you've talked about how we go about estimating or determining the absolute date of remains. This individual would have been determined through, and again, if you're thinking, let's think back here, we're using C14, carbon 14. That's what we're using for organic materials. And I'm going to do this as we go along. I'll put some notes on the side to help you take some notes as well, some of the important information. And our calibrated date for this individual is approximately 2,000 years before the present. Now, fairly recently, these remains, they had been sent off to McMaster University for some stable isotope analysis. I'll write that up at the top as well. Okay, so stable isotope analysis. And by looking at stable isotopes over time, and carbon 14, that's an isotope, and because it decays, it's not stable. But in contrast, for example, C4, C5, C12, C13, there's a whole list here. They are stable over time. And by analyzing that, we're able to figure out a little bit about these individuals. Now, what they were interested in was determining where these people came from. So we have people living in England, these remains, that's where they're coming from, and they were calibrated to 2,000 years before the present. But how do we know that they're ancient Rome, that they came from ancient Rome, the city? Well, what they did is they engaged in the stable isotope analysis, and in doing so, they were able to determine the individual's water source. the water isotopes from where they grew up. And by doing that, they knew that the isotopes that are found in this individual's bones, it's doesn't indicate that the person came from England. Instead, it's more consistent with isotopes that are found in water in Rome. So we know that this individual came from Rome. And one of the things that we know about London at this point of time, it wasn't just your traditional Italian Roman individuals. This was a very multicultural society. And again, we're getting at that information through the stable isotope analysis. Now, McMaster, they're going through this stable isotope analysis, and they came across some really interesting information. Let's move to the next slide. Right, so here we're looking at the same individual. We're looking at a variation in the museum exhibit, and I'm going to go up to the top. Variation. I've used the two images just kind of as a lead in to the idea of variation. It does exist. You know, this is the one thing that Charles Darwin, he had pointed out in his Origin of Species. Right, the Origin of Species, he talked about that variation that we see with all animals, and he said that if these variations that appear over time that are totally random and natural, if they provide an adaptive advantage, I'm going to squeeze that in. If they provide an adaptive advantage, they'll be selected for through that process called natural selection. And ultimately, this leads to the idea of evolution. Now, variation, it doesn't just exist among tortoise, horse, dogs. It also exists among humans as well. And for a very long time, we've been told that there's, you know, when we look at humans, we either have males, and from a very early age, we're put into a male box, or we're put into a female box. And we've also had a lot of individuals begin to challenge this. And this is why I think this case is particularly interesting, because it does provide us with archaeological insight into the notion that variation exists among males and females in a way that doesn't permit this clear binary opposition between us. In fact, I think when we look at sex, it's probably better described as a continuum from individuals who are very textbook male, individuals who are very textbook female, and then we have all kinds of categories in between, and people fall somewhere on that line depending upon a number of different factors, even one's own identity in the way that they perceive themselves. So we've come a long way from saying that there are just males and females. Now, looking at the skeletal remains, when they were excavated, archaeologists, bioarchaeologists, they said this individual is clearly female. What I've highlighted here, if we look at the box with a little red line, okay, so here's the box and the little red line up top, that's on the greater sciatic notch. And that's one of the areas that provides us with some insight into the individual's skeletal sex. Now, like I said, McMaster University, they took a look at these remains. They were analyzing for stable isotopes, and they were also looking at the chromosomes, looking at DNA, extracting that stuff. McMaster, awesome at doing that. And what they said is ultimately, this individual sex is indeterminate. And people say, what are you talking about? It's clearly a female when we look at that greater sciatic notch and other indicators on the body. And the other thing I'll point out here, if you look down at the bottom, not only do we have the skeletal remains, but we also have jewelry, also indicative of a female. Now, one of the things I want to point out here is jewelry and clothing doesn't indicate sex. It does indicate gender, one's own perception or identity of who they are. Now, what McMaster points out, really the same thing, is we need to understand that variation always exists. It exists among all life on Earth. When they looked at the skeletal sex, as pointed out, The individual is female, however, the chromosomes are not. The chromosomal sex of this individual is male. So here, genetically, the individual is male from a chromosomal level. However, the skeletal sex indicates female. And when we look at some of the artifacts associated with that, we could say that the individual did identify, depending upon the time frame and what those artifacts mean, identified as a female even though they were genetically male. So very interesting work, because this challenges that whole notion of a very clear binary opposition between a male and a female. We know that there's intersexed individuals, and here we say, yeah, in these situations, we're going to call this individual indeterminate. We cannot make a clear estimation based upon what we're seeing because, well, we can't talk to the individual and get further information from them. So what is sex? Well, one of the things to remember, it is not gender, okay? Gender is something very different, and I know it's confusing. We look at doctors' forums and they ask us what our gender is. Sex, this refers to biology. When we talk about gender, we're referring to something different. Gender refers to a social and psychological construction. Okay, so it's perfectly possible to be biologically, genetically male, and when it comes to what one identifies as their gender, socially, psychologically, that is something that they construct based upon how they feel ultimately. So we can say here, number one, when we're looking at sex, and we're estimating sex on the skeletal level and the genetic level, this stuff is related to reproductive functions. Okay, so the differences that we see between males and females when it comes to the pelvic girdle, they are fundamentally different because males do not carry babies, females do, and we need a very different anatomy. So our primary sex features, okay, so those refer to, for example, the one that involves carrying a baby. Okay, so we'll just say carrying a baby. We need a very different hip structure, not only for carrying that baby, but also for delivery. Other primary sex features that wouldn't be skeletal, for example, would relate to one's breasts. So females, they have different breasts than males, and that's related to, again, that reproductive function of providing milk for the offspring. And we see that consistently across species. So reproductive functions, other soft tissue, for example, genitalia, we can't see that on the skeletal record. So we do need to really refer to these types of features that have everything to do with carrying a baby and birth. And we also need to keep in mind that not everybody is created the same either. When we look at delivery, not all women have the pelvic girdle that allows for a delivery without complications. And again, that's because we do have variation among us. So here we can look at sex from a number of different perspectives. Number one, it could refer to our chromosomes. So humans, we have 46 chromosomes. We get 23 from our mother, and we get 23 from our father. Okay, so 23 from mom, 23 from dad. Mom always passes on X chromosomes. Dad can pass on an X or a Y chromosome. So an individual that is XX, this is what we would define anthropologically. We don't use that traditional sign for female. men are from Mars and females are from Venus. So the story goes. So we don't use that sign for Venus. Instead, we use just a circle. Okay, so XX is a female, a circle. I'll just put an F over top so we know that, so we can remember. And if an individual is born 23rd chromosomes, they're X and they are Y. There we go. That individual is, here we use a triangle, that individual is chromosomally male. That's one thing, chromosomes. We can also look at it on the genetic level, and here is, we're not going to go into what people have previously defined as disorders, genetic disorders. We're going to just say that there is genetic variation. We don't want to use that word disorder anymore, and especially if we're looking at this from an evolutionary perspective. So there's chromosomal sex, genetic sex, there's skeletal sex, and sometimes these do not align. And we also have identity, or one's own conception of who they are. Now, some of this stuff we can get at when we're looking at skeletal remains. So everything that has to do with those skeletal remains, if we're engaging in an anthroposcopic, right? We've talked about that earlier. You've been introduced to that. So that visual inspection of skeletal remains, the anthroposcopic analysis is going to give us some insight. We can't get to this identity through skeletal remains. We could use artifacts that are found in association. That could help us get to that idea of gender, how the individual identifies. And if we went and began to look at the bones on that microscopic level, we could get at the chromosomal or the genetic sex of the individual. But quite often bioarchaeologists, they just look at that skeletal sex, and that's what they make their assessments on. And the same thing would be true of forensic anthropologists. They look at the skeletal sex, they'll engage in anthroposcopic analysis and also anthropometric analyses as well, so measurements. When estimating sex, we need to keep in mind that there are physical differences between males and females depending upon, well, what species we belong to. The word that we use here is sexual dimorphism. I'm sorry, that's a big, ugly word. This just means physical differences that are related to, I lost my A there, we'll put it back in, related to sex. Okay, so asking you here, which orangutan is male, which one is female? I'll give you a second, take a look at the two. Clearly, the individual over here, the orangutan, is male. This one is female. There's a lot of sexual dimorphism between them. In other words, when we look at the male, we see a very different cranium, for example. The weight, so the skull, we see massive differences. And just look at how this is flaring out on the male, whereas we don't see that same type of facial flare. The other thing that we have, if we looked at the skull, would be a sagittal crest, the bone. It comes up. This individual doesn't. It's just nice and smooth. So we see those differences with respect to the skull. We also see those differences with respect to ultimate body size. There are some huge differences, and if you think about the differences between a male gorilla, for example, and a female gorilla, a male is nearly double the size and weight as a female. In contrast, when we look at humans, we don't see the same type of dimorphism. And if we looked at, for example, cockatoos, a Goffin cockatoo, it's almost impossible to tell the difference between a male and a female. If we look at a peacock, we immediately know the differences between a male and a female. The male has that large fan tail. That is a type of sexual dimorphism. So species, we do see those differences in them as well. looking at two humans here. So we clearly knew which was the male and the female gorilla. I'm going to give you a second. Which one do you think is male and which one is female? Okay, so this is part of a study from the University of Montreal. And if you said female over here and male over here, you responded in the same way that the people in the study did. And some of the differences that we see between these individuals, for example, the distance between the individual's lips and their nose, the distance between one's eyes. There's a few things here, but it's the same person. Okay, they're both males, and what they're trying to illustrate is, we just change a few features on an individual, and they begin to take on features that individuals will judge as either male or female. Now, in the case of forensic anthropology and bioarchaeology, we don't have tissue to judge, but we do have distances, and we're not going to go into that anthropometric analyses. We're going to keep it to anthroposcopic here. Anthropometrics, it's just going to be something that you would cover at university, so we're not going to do that here. We also need to keep in mind, as I've already pointed out, when we're estimating sex, we need to do this with post-pubescent individuals or people who have gone through puberty. So, for example, if I ask you, out of these two, who's the female? Who's the male? Is there a male? Is there a female? So looking at an infant, this is very difficult to do because we don't have those features that are being initiated by puberty that are ultimately related to reproduction. Okay, so that stuff, those hormones, they haven't initiated. If we're really curious here, we could say, oh, a little bit of anthropological or social insight. I see a little bit of clothing, perhaps, so I'm going to say that that's pink and therefore that's a female. Well, we need to be careful because pink doesn't necessarily equal female. In our culture, yes, when children are born, they dress little girls up in pink and they put them into that female gender box, feminine. Okay, so that's the gender. Okay, so you would be feminine by wearing pink. And when we have a little boy, we like to dress them up in blue and we put them into a mailbox. That could get us into trouble. Don't put anybody into a mailbox in that sense that we're seeing there. The Postal Service might become upset with us. Either way, okay, bad joke. Either way here, we need to be careful about those assumptions. Just because somebody's wearing blue, that does not mean that they're equal to male. And the other things that we also see have to do with even the way that we've been taught to behave or that we've been socialized to behave over time, and people make those judgments. So here in anthropology, we can't make those judgments. And when we don't know, and in your case study that you're going to do, if you can't determine it, that's fine. Keep in mind that a forensic anthropologist, if they're in court and they're testifying, they can't say something that they're not certain of or they're pretty darn certain of when it comes to their estimation. And it's okay to say that I think that this individual, the sex is indeterminate because of A, B, and C. So it's okay, I'm going to say here, to not have a conclusion when it comes to a formal estimation. Because sometimes we can't have that formal estimation other than saying indeterminate. Yeah, I'm losing my N. Indeterminate, but the thing is, what I'm looking for, and we always want to see, is it's indeterminate or we can't determine sex because of A, B, C, and D. So we're going through that support, we're giving an analysis, and that ultimately is the work that we're looking for. Now, while you might think that you're looking at a bush baby, a type of prosimian, like a monkey, this is not a monkey. And this is, again, another good example of why we cannot determine sex when we're looking at post-pubescent individuals. These are two humans. Your guess is as good as mine, which is male, which is female. We don't know. It cannot be determined. So in contrast, normally when we're face to face now, here's where I'm going to turn it back to the class. I'm going to say, okay, you tell me which one of these individuals, A or B, is male or female. Okay, so I'll give you a second and take a look at the individuals. The first thing that you're going to see when we're comparing A to B is we're seeing some dimorphism. So we're seeing some differences in size. So the cranium of B is clearly larger than the cranium of A. The other thing that we see here pretty quickly is something that's happening in the forehead, so that the frontal bone. So remember, here's our frontal bone. There's our frontal bone. And this individual's frontal bone, it has bossing. You can see that it's kind of got that straightish, bulbish forehead, whereas this individual's forehead is sloping back further. That's one of the differences that we see between males and females. In addition to that, if we look at this space right in between the eyes, okay, this is an area where we see something called the gallbella. And here we see a gallbella, a very pronounced gallbella or supraorbital brow ridge. Okay, so larger brow ridges. If we progress down a little bit further, right to this area, we can look at the mastoid process. And we're seeing a larger mastoid process on B, a smaller mastoid process on A. And again, B being more consistent with male. The other thing that we can look at is this this orbital ridge margin right here and right here, and we can see that this margin is larger. That bone, it's thicker. So think of a cup, for example, that margin on a glass. If it's china, it's going to be smaller, or a wine glass, stemware. So that's a margin right here. And in contrast, your old Tim Hortons cups, when we went and sat down, they're much more robust, larger. The margin is thicker. So here we're seeing a much wider margin, and in contrast, A, we're seeing a thinner margin, not margarine. I prefer butter. Either way, that allows us to say that A is consistent with female. And I'm going to teach you how to rate this, and B is more consistent with male. And if that was your guess from the start when we're looking at it, you're absolutely correct. Now you're just going to have to explain why you know this stuff. Okay, let's move on. All right, so a little bit of review, and now linking that up to sexing a skeleton, or estimating the sex of a skeleton. Anthroposcopic analysis. You learned this at the beginning of the course. This refers to visual inspection. So here we have visual inspection of a known male and female features, and that's what we're after. We know that the skull, for example, it provides us with excellent landmarks and features that we can use for anthroposcopic analyses, and the same thing is true of the pelvic girdle. If you're looking at any single bone aside from the skull and the pelvic girdle, ultimately you've got a 50% chance of being correct on the basis of any single bone, being either a male or a female. The other method that was discussed, and this was traced back to Hooton, is that anthropometric or a biometric analyses. So this refers to measurement. Okay, measurement of long bones, for example. We see differences, and ultimately because, well, males, I was to kind of draw in a male here. In general, males are taller than females, are they not? Okay, so we can say that if a male is generally taller than a female, if we look at that, I'll draw in a femur here. Again, my marker's playing havoc with me today. I apologize. So what we can expect is that femur there, it's going to be larger than the femur that we're going to see in a female. Okay, so we have known averages. We'll talk a little bit about that in a second. And on the basis of that, we can also use that to help us understand whether or not the individual is consistent with a male or a female. We want as much information as possible. All right, so the male femur, on average, it's about 480 mils, so we'll put a triangle above. So right in this general area, we're looking at people that are probably about 5 feet 11 inches to about 6 feet 1 inch. We'll say that's not important. That's just for us to understand this. But we know that there are individuals who are much taller. Some individuals, they're 6 feet 4 inches. Some individuals, 6 feet 8 inches. Some individuals, 7 feet plus. Okay, but that tapers off as we go along, and we also have individuals, males that are 5 feet, that are 4 feet, and that tapers off as well. Now we have to superimpose here the female average bell curved as well. Okay, and we have an area that's overlapping, a number of areas that will overlap. So the problem becomes when we look at individuals that do fit into this category, we can't simply assume that because an individual has a femur that's 430 millimeters, that it's necessarily a female. It might very easily be a male who is shorter. The same thing is true of a female who would fit into this category right around here, who has 480 millimeter femur. We could assume, well, the person's tall, they're over 6 feet if we're finding a femur that's in this area, but that doesn't necessarily mean it's a male, because again, it tapers off with that bell curve. Think of it in terms of grades. Not everybody has an average of 75. Some people have an average of 80. Some people have an average of 60 and 50 and downwards. So like I said, the same thing applies when we're looking at this dimorphism. Now, there's a number of factors that will affect this as well. Okay, so we know that some individuals are tall, some individuals are short. It makes it difficult for us to just look at the length of a femur. What other factors do you think affects our ability to determine sex by stature estimation, if we're saying tall versus short? Is there anything else out there? If we think again about time, for example, and we go back in time, I'm going to ask you, have people always been the same height? So this is going to be important if we're doing bioarchaeology, or if we're going a little bit further back in time prior to a contemporary era right now, because people's height, they do change. What about ancestry? Do you think that affects a person's height as well? If you're saying, yeah, there's certain groups of people who tend to be very tall, for example, that come from Nordic areas. Yes, very tall individuals. And as we move through other areas of the world, we find individuals who are much shorter. And now all of that's starting to change in modern times, but we need to take this into consideration. So let's look at a couple of examples here really quickly. All right, so this is how we measure a femur. It's measured in a measuring box, a sliding box, and here we see the individual. Okay, so I'll use a red one. It's a little bit easier to see right there. 38 centimeters. We'll convert that 380 millimeters. And how are we going to determine sex on that? Well, we said 480 was a male. We're seeing 380. We're going to definitely say female, correct? Let's write that in there. We're looking at this individual. If you're shaking your head saying, no, we can't make that conclusion, you're absolutely correct, because time is going to have a play in this. Now let's look at height over time on our next slide. All right, so what we're seeing here, human heights over the long run. Average human height in Eastern Mediterranean from the Upper Paleolithic. So that's before 16,000 years before the present, or BCE. I'm going to correct that. We don't like to use before Christ because that's biased towards one religion. So we're going to say BCE through 1980. And if you thought that people were shorter in the past, you're right and wrong. If we're going to go back to 16,000 BCE, the average height of people, I'm going to go and highlight this in green, compared to modern males today, they were taller. What ended up taking place over time with the invention of agriculture, so these changes that we're seeing, for example, here we're seeing a decrease in height until we get about 12,000 years. There are some really drastic climatic changes that are taking place that are having an impact upon availability of food. And then farming's invented. Okay, so right around here, farming is invented. And contrary to what most people think, they think that farming was this huge development in human history that helped us. Actually, our heights plummeted because of the availability of food. And only after 4000 BCE did we start to see increases, a little bit of a plateau, an increase as we just start to come up to, this is probably around 200 BCE, and then ups and downs until we finally reach our modern height. And females are pretty much the same here. We see that same decrease in height over time. Again, availability of food, and now we're seeing females start to get to some of the height that they were previously. So here, when we look at the remains of people that came from 16,000 years before the present, and we estimate their height based upon bone lengths, we know that they were taller. There's no question about that. Okay, how Canadians have grown over time. If we look at the last 100 years from 2014 back, 1914, on average, women were 5 feet and 2 inches, and in 2014, they'd increased 2 inches average height. The same trends are true of males. We're seeing about a 3-inch difference between 1914 and 2014, 5'7" to 5'10". Now, the other thing that also needs to be taken in consideration, it's not just time, but as I've pointed out, ancestry. So here we're looking at a number of different heights, individuals represented by flags. For example, Bolivia, the average height currently is about 4 feet 8 inches, and the Netherlands, we're looking at the average height of women being 5 feet and 7 inches, and a number of individual averages between based upon country of origin. So again, determining an individual's sex on the basis of anthropometric analysis, and just that, it is fraught with a number of difficulties. So we want to gather as much information as we can to build a case, the strongest case that we can for either male, female, or indeterminate. Okay, so that's where I'm going to leave this brief introductory lecture on estimating sex, our introduction. Where we're going to pick up is estimating sex, and first we're going to be looking at the skull, and this is a pretty important area because it gives us about 80 to 90% accuracy. And then our final lecture in this area, we're going to be taking a look at the pelvic girdle, and that's going to give us 95 to 100% accuracy. Okay, if you're interested in taking a break, like I said, that's where I'm going to leave it. If you're going to plug through this stuff, I'll see you in a couple of minutes. All right, take care.